So I'd like to introduce our next speaker, Jonathan Udell. As long as I'm giving Celera anecdotes, I wanted to mention that Jonathan is part of a very curious and I think not well-known part of Celera's history, and Sridhar actually set it up perfectly. So after the genome was sequenced, I think there was some casting about for what Celera does next. And I have no idea how the decision was made, but the decision was that we would be working on cancer immunotherapy. And this was in some ways, a very far-sighted decision because it was really a pie-in-the-sky project then. And today, cancer immunotherapy is the hottest thing in cancer. But it was too far-sighted because it was about a decade after Celera went away that it actually, the major problems, checkpoint inhibitors, were solved, and it actually became a useful technology. But it led to some cool science, and it let those of us who are there say that we were doing cancer immunotherapy before it was cool. So we got to work with Jonathan and Steve Hoffman and others, and I hope you, that is, of course, one very small part of what he does. He's really a giant of the field of immunology. And of course, it was recognized we couldn't do this without an actual immunologist. And we're really happy to work with him, even though we never cured any cancer. So thank you.
Well, you could see where Celera was heading if they had to consult with me to make money. So that was certainly a harbinger. Okay, so now for something completely different. I know many of you you know biology only as much as you get sick or are worried about COVID and relatives, et cetera. But I'm gonna tell you some immunology. There'll be like no data. If there's a little bit, I'll describe simply what it is. And we're gonna talk about the immunopeptidome. So very good introduction. Immunopeptidome is something you need to know if you want to specifically target cancers with T cells. I have a subtitle. There we go. Making drips great again. And so you're wondering what the fuck is a drip? I will tell you what a drip is. It's not a person, although it's both actually, right? Okay, so T cell immunity. There are two kinds of T cells. You have your helper T cells, which is what get boosted when you get the COVID vaccine. You need the helper T cells to make antibodies. Without helper T cells, you don't make a good antibody response. There's another kind of T cells called the CD8 T cells. CD8 is just a cell surface protein. That's a differentiation marker that's very convenient. And these guys originally were called cytotoxic T cells. This is what won a Nobel Prize for Zinkernagel and Doherty in 1996 because these cells play a really, really important role in viral infections. They don't prevent viral infections, but they make sure you don't die from them. COVID would have been much, much worse if SARS-CoV-2 wasn't related to other coronaviruses that we get routinely. There were four circulating coronaviruses. We have a fifth, and now some of the other ones are probably going away. The T cell determinants that are recognized by the helpers and the killers are conserved, many of them. So we were not a completely naive population. Think about that. 1 million dead Americans with vaccination, right? Not all vaccinated, of course. The number would have been much, much higher. Probably would have been 1918 flu-like had there been no cross-reactive immunity. Okay, but there was some. So these are really important. And I've spent a good chunk of my career studying CD8 T cells. What do they recognize? They have a clonally restricted receptor called the T cell receptor, or TCR. You have, 10 million plus different clones floating around in your blood right now. Most of them will have completely unrequited love in their whole careers as T cells. They'll never find a target cell. But the lucky few ones find a target cell and then they proliferate like crazy. We have a really cool paper in eLife where we actually are counting. You guys are quantitative. We count the proteins in the cell and the ribosomes. And we looked at T cells because they pose a really intriguing biological question. They can divide every 6 hours when they're cranking. How the hell can they do that? Right? That's a lot of protein synthesis. And it's a cool paper because we have a paradox. We try to do the numbers as carefully as we can, and this is not the numbers you guys do. This is what you learned in first grade. Immunologists are capable of that, most of us, which is just counting. We count proteins, we count ribosomes, we can measure the rates of translation. They shouldn't be able to divide as fast as they can, and yet they do. So we didn't answer that paradox. Okay, but that's something else. So I'm going to talk about Wait, I gotta go back. I'm sorry. I'm gonna fly back in here. The origami's coming back. What the TCR recognizes something called an MHC class 1 molecule. This is a really cool molecule. This molecule evolved to take a peptide that's made inside the cell that's, say, 8 amino acids to 12 amino acids, and it carries it out from inside the cell to the cell surface. It's a phenomenally sensitive system. A T cell can kill a cell expressing 1 or 2 copies of the peptide, right? Out of 100,000. TCR sorts through all these and then makes literally a life or death decision whether to kill the target cell. So these are really important in viruses, and it turns out Celera was ahead of the curve, absolutely. They are also very important in cancer. The way the checkpoint inhibitors work, at least 50% and probably more, is by unleashing the T cells. Cancers are not stupid, right? By the time someone shows up in a doctor's office with a cancer that's clinically presenting, the cancer has evolved to evade the immune system. There was a very long debate in immunology whether cancer immunosurveillance even exists. It does. One of my college roommates is living proof of that. He would be dead. He presented with like 4 pounds of melanoma. He was one of the poster children for checkpoint therapy. Within 6 months, there was no detectable cancer. So when it works, it is unbelievable. Dead people are walking. Field of antigen processing, fantastic field I helped get started in the 1980s. Lots of great cell biology. There's a pathway, there are textbooks. All the rules are breakable because it's biology, it's not physics or math, okay? So everything you read is probably a little bit true-ish, right? It's one thing you guys need to know if you haven't already learned about biology. Things that look like facts, at very best, they're sometimes facts. And there are always exceptions, always. And this is just a classic example. Everything I'm showing here is way oversimplified. Basically, you have a protein. It's a great cartoon. It's a chicken because we all work on a chicken protein and chicken peptide, which is stupid, but we do. It's a very useful system. The protein gets degraded by something called the proteasome, which is this, Cuisinart for cells that could basically degrade every protein in the cell into little bits. Some of the bits make it into something called the endoplasmic reticulum, which is the inside part of the cell, where you assemble things that are destined for the outside part of the cell. And what happens here is the MHC class 1 molecule is assembling with the peptide that's going to carry out to the cell surface, where the T cell is going to decide whether to beat the cell with the pipe or not, okay? So this is the class 1 antigen processing system. The repertoire of all the peptides that are presented, and there are millions, is called the immunopeptidome. Where does that word come from? It comes from this paper, because we invented this. In helping destroy Celera as a financial enterprise, we invented the word immunopeptidome. And this is a really great paper, and thank God that somehow you guys found me. I don't think I've ever had as much fun in my career, because to be honest, this was my take on it. Steve Hoffman was clueless, right? Completely clueless. You guys, you had all these incredible brains, and you needed a guiding rudder. And I have, as Soren knows, anyone who knows me, I have more ideas than I could ever follow, like most scientists. And this was working with the dream team. It was just absolutely the dream team. You guys were so smart. I would have an idea. 2 weeks later, there it would be. And we were the first ones to apply informatics to this question of where do the peptides come from and what is the evolution? And I think my favorite experiment in the paper, the question was, okay, did the proteome, did the genome evolve to favor or disfavor peptides from being presented? And this was pre-Celera, but there were already decent algorithms. They've gotten much better for predicting. It's one of the early examples of where machine learning worked in biology for predicting peptides that would bind to class 1. These rules were pretty good then. They're a little bit better now, but they were good. So we did this study where we could predict peptides and whether there was evolution for binding or not binding class 1 molecules. And my favorite organism was the Vent organism. Right? We took one of these organisms that evolved at the bottom of the ocean at huge pressures and gigantic temperatures, and it turns out they were no more or less liable to have a binding peptide than we are. Right? So there was no selection for or against peptides. That was what I love about informatics is when you answer a question like that, it is definitive. Everything else I do, it's not definitive. Because even if your evidence looks great, as a biologist, experimentalist, you're always worried. Did we do this? Did we do that? Did we pick the wrong salt concentration? Did we not? Why didn't we do this control? you do something with informatics, okay, maybe it's completely wrong, but you don't have to worry about the method, right? Just maybe you have to worry about your assumptions, but not the method. Okay, so I got to know Soren, and we had, for a while, we had a pretty close relationship because he was a cool guy, incredibly brilliant, and I had lots of ideas about what one could do with informatics, and he invited me to Brown, just downstairs, and at that time I was already giving this talk I give for young scientists on how to succeed in science, and I gave it to a group of Soren's people, and I found out it was actually useful. Right, so that's for an informaticist, that was really quite an amazing thing. So thank you for that last night. I didn't realize that. Yeah, and it's been online ever since. And I think a lot of kids have seen this video. So thank you, Soren. And I hope Brown keeps it posted, for forever, basically. I'm still giving the same talk, only it's not as good because I'm really slowing down.
Derek was the one who put it.
I still manage that account.
Thank you, Derek. All right, we have to keep Derek alive. Yeah.
Derek.
Yeah. Make Derek great again. Yeah. Okay, and that talk led to a couple of essays that I'm happy to send to you. They're, I think these have been downloaded hundreds of thousands of times about how to succeed in science. And then I eventually took a sabbatical where I went there not to write the book, and eventually I wrote it. Right. So there's a book. I wrote this as a government employee. You're not allowed to set a book at 0 on Amazon, right? So I'm actually breaking the law because it's for 99 cents. But if you don't want to pay 99 cents, email me and I will send you a link to a Google folder where you can download the book in an e-format, okay? And, it's a useful book. It's very practical. Like the books I read when I was a student, they just pissed me off. There's a famous one by a guy named Peter Medawar. The title of it could have been, I'm smart and you're stupid, right? So my book is not like that. It's just, what does it take to get through graduate school? What does it take to do an experiment? It's very pragmatic. Okay, so what are drips? The drip hypothesis from 1996 was proposed by me basically to explain a curious observation. You take cells, you infect them with a virus. You have a trick for stopping protein synthesis various times, just a drug, okay? You do that, you find out 30 minutes after the virus has attached to the cell is enough time to make enough protein that the T cells can kill the virus-infected cell. Whoa, that's really fast. Makes sense. Viruses are very fast. SARS, not so fast. Some viruses can replicate in 4 hours. SARS-CoV-2 in vitro anyway takes a day or two, but still fast, right? So that is really fast. Okay, so this part didn't make sense. The peptide, even at that time we knew what the peptide was, it comes from a protein whose turnover you really can't measure because the cells die. So the average cellular protein has a half-life of a day or so, right, on average, across the whole proteome. For viruses, the proteins are at least that stable on average. They're really stable proteins, okay? Another problem, you have, in 30 minutes, you have maybe a few thousand copies of viral proteins. How many copies of host proteins in a single cell? 2 to 3 billion, something like that, for an average size cell, billion. So 100,000 class 1 molecules, you guys can do the math in your heads, that ain't gonna work. If we're gonna wait for degradation, there's no way. Right, the kinetics they're not going to allow it. So, hypothesis is that to enable immunosurveillance, a virus's peptides derive from something that we call a DRIP, which is a rapidly degraded, defective ribosomal product. So I didn't want to dignify the substrate, as a protein because we didn't know what they are. We still don't know what they are. And I wanted to leave open the possibility that it was just like a shorter peptide. Right? So I called it something that wasn't protein in the name, a defective ribosomal product because it is a product of the ribosome, right? All of you know the ribosome is the thing in the cells. To me, evolution's greatest achievement, making this machine that produces all of the proteins on Earth, something called the ribosome, right? Every cell has to have them. So here's the idea, right? So here's another cartoon from a friend of mine named Eric Reitz. We're making the chickens, the model protein, they're coming off. Some of them are mistakes. Maybe they started in the wrong place. Maybe they finished in the wrong place. Maybe there was a frame shift. These are headless chickens. These protein synthesis errors in protein folding, which is not perfect, very imperfect, right? Sometimes. All these guys are going to be triaged to the proteasome. We're going to make peptides, and these are going to be presented to the immune system. Okay, so that's the idea. That's the simple idea. And for viruses, this turns out to be really, really true. So this is the only data I'll show you. So here, this is mass spectrometry, not by my lab, by a very good lab in Australia, a guy named Tony Purcell. And this is influenza virus, infected cells. We're looking at influenza virus proteins. The names don't matter. The red is the protein, okay? The grayish thing are the peptides from the protein. Look at this. Sometimes they can detect the peptides before the protein. As I said, all of these proteins are stable. This protein and, is that any there? And this protein are infinitely stable because they're exported, right? So they go outside of the cell, so they don't even count. So in that case, the peptides had to come from drips. But even from the other ones that are not exported, that are staying in the cytosol, in the nucleus, the peptides are being made basically co-translationally. So this is confirmation of the model. Okay, and this has also been done for vaccinia, but for no other viruses yet. The mass spectrometry is not easy. And you have to be a lab dedicated to it, and it's costly.
Y-axis on the previous slide?
Sorry, I should have explained. This is just a relative amount. Yeah, and it's not super quantitative either, which is one of the problems. If you want to quantitate the peptides or the proteins, not so bad. Peptides, you have to make a standard curve with a known peptide.
And do these peptides come from throughout the protein or only from the base?
No, they come from, that was something we actually looked in this original immunopeptidomics paper. They come from throughout. There's some biases that we still haven't figured out, but more or less it's from the whole protein. Okay, so what are they? When you make a full-length protein, what are the peptides, or when you have a full-length gene? So everything you could think of, every possible error in literally translating information from the original nucleic acid to the protein. It's proven difficult to nail down biochemically what they are, but after 27 years, here are 3 examples of defined influenza virus drips in ascending order of craziness. Most of these just found in the last 5 years, some in the last couple of years. One is in one of the gene products, it's a +1 reading frame, starts on a methionine, peptide starts there, 14 residues, right? So something that small, anything less than 50 residues is probably not going to fold. So unless it finds a partner, it's going to be degraded. Something that's 14, it's not impossible that it has another biological activity. There are some peptides that small that bind something that have activity, but this is probably just a mistake by flu, okay, influenza virus. There's another one, it's a model peptide, but still tells you what can happen. This is a crazy one. It's in a +1 reading frame, it's after a stop codon, and it's initiated not by AUG, but CUG. So what antigen processing tells you is that all the things you think you know about what it takes to make a protein, you have to expand them. Do you mostly make proteins on AUGs? You do. But actually that rule is really coming into question now. There's more, if you're just counting gene products, not abundance of proteins, more than half of the gene products, way more, are initiating on non-AUGs than AUG, right? So that rule is really not a rule. And then here's the craziest one. We published one example, we now have 5 other examples where we are making peptides from the negative strand of a negative strand virus. Okay, so what a negative strand means is it's non-coding. So whenever I use the word non-coding or UTR, I have to make air quotes. Because a lot of the things we think are not translated, they are translated. And I'm not telling you it's abundant translation, but I'm telling you the immune system thinks it's interesting. And the immune system can use this for immunosurveillance. Okay, what is the overall contribution of drips to the cancer immunopeptidome? So we had a really good collaboration with a very good friend of mine at NIH called Lou Stout. Lou is the world's expert on human B-cell lymphomas. And we took three of the human lines that Lou has developed. We did ribosome profiling. This is a method worked out by Jonathan Weissman. Incredibly difficult, incredibly powerful, right? This is a method after $10,000 and with a really good postdoc and doing it 6 times, you can figure out everything a cell or a group of cells is translating. It's an amazing experiment, amazing method. And then we had a very good collaboration with an excellent informatics and peptidomics group in University of Montreal. And this was a really nice paper in Cell Reports. My lab did the ribosome profiling. The other guys did everything else, including the bioinformatics, which in this paper I thought was quite amazing. So the RiboSeq data, this revealed 181,811 non-annotated proteins, in addition to the 26,000 plus canonical proteins. So Craig was saying the other day that the 400,000 number was wrong, but maybe not as wrong as you thought. And not for reasons you ever could have anticipated. Because these are things that, again, by definition, they are non-canonical, right? So, but there are a lot of them, and this is not the only paper. Original paper from Weissman, they were already showing there were tens of thousands of non-annotated genes.
These are short peptides or actual proteins?
There are all sorts of things, right? But things you think are UTRs, they're not necessarily. A lot of them are translated, right? And then this is what we're detecting as well. And even though you're getting what seems like a lot of reads, right? 500 million reads, that's 100 cells worth of ribosomes. It's nothing. So God knows in a cancer what exactly is being translated, right? It's a lot of things. So mass spec, not as sensitive as RNA, DNA-based methods. This is a good group, 14,500 proteins they're detecting. 17% are non-canonical out of our 181,000. So 28% are splicing variants. 72% are cryptic, UTRs, frame shifts. The cryptic proteins are predicted by bioinformatic algorithms to be more disordered, unstable. They're drippier, according to us. And this we can do per translation event, because that's what we're counting by ribosome profiling. They are more likely to create a detectable peptide, right? So that all kind of makes sense. Okay, so thank you, Bill Barr. We're breaking the law here, right? How does the immunopeptidome circumvent the law of mass action to prevent the most abundant proteins from dominating the immunopeptidome? 250 proteins account for 75% of the proteome, 250. These are the housekeeping proteins, molecular chaperones, ribosomes, translation factors. The most dominant proteins are a huge fraction of the proteome. If it was just a come-as-you-go system, the immunopeptidome would be dominated by them, and it's not. And you wouldn't be able to see viruses. So we're breaking the law of mass action in terms of not every protein that's made, they're all going to be degraded, winds up with an equal chance of being a peptide that's presented. How does that happen? We had an idea a long time ago, 20 plus years ago, a real unicorn for my lab. The immunoribosome hypothesis is that a subset of ribosomes generates substrates at high efficiency for immunosurveillance. So the idea is you have 3 million ribosomes, maybe 10,000 of them are actually really good at making peptides for some cell biological reason. That was the idea, right? So the immunoribosome makes a drip, which goes right to the proteasome, which goes right to the transporter, which goes right to class 1. That would be a model of something that's what we call channeling in cells, right? Many enzymes are known to be channeled. You don't randomly have a substrate going and a product going from one enzyme to the next in a metabolic chain. You organize the enzyme so it's more efficient. That would be the idea. Okay, so it was proposed in 2002, and not widely for you guys, but widely for my tiny little field. My 15 colleagues who knew about it thought it was a stupid idea, right? That was widely ridiculed for this field, right? Okay. But what I love about science, which is my arrogant name of the book I wrote, is truth wins, right? I started as a history major as an undergraduate, and it was like whoever was the best bullshitter won the argument. And in science, I'm not saying the truth, I think, as a biologist, is something we approach asymptotically. We never get there, but you have evidence, right? So if you're right in the end, you could say to the world, fuck you. right? That's the great thing about being a bench scientist. If you're right, you can show it eventually, or somebody can, right? Hopefully your career won't be over by that time, but that's one of the advantages of being in the intramural program in NIH. We're not like Elliot Ness's untouchables, but close, right? So it's hard to fire us, right? So it's good to be there. You can have big ideas and just ignore people when they criticize you. You don't have to convince the study section. It's not a completely stupid idea.
That's why ESTs work there.
Yes. Okay, and eventually I got lucky. I got a great person. My lab, postdoc named JJ Wei, who's now at Merck. And JJ was the unicorn. And what JJ did was knock down every ribosomal protein and say, does this affect antigen presentation? And it did, okay? We could upregulate antigen presentation. We can downroad antigen presentation. It had nothing to do with how much protein was being made. Okay, we did this, really nice. We had no idea how it worked. We had no idea how important it was. But is this relevant? It turns out we got really, really lucky at the end of this, which is why the paper's in Molecular Cell, because these knockdowns of ribosomal proteins are what cancers do. Very few cancers that don't upregulate their ribosomes or downregulate or even get rid of some of ribosomal protein. So the ribosome is mostly an RNA machine, right? And the RNA itself can be a shitty ribosome, but the proteins make it a lot better. There are 80 of them. Okay, and we found out you can knock down 20 of them and have an influence on peptides. And it also turns out that a friend of mine at NIH named Nick Restifo had just published a paper in Nature where they did the first CRISPR screen for how tumor cells can evade cytotoxic T cells. And oh my God, one of them, where's the smoking gun? It's pointed the wrong way, so the slide got screwed up. But RPL23 is a ribosomal protein, this guy up there, right? So there it was. Could this be relevant? JJ started going to their lab meetings and doing experiments and hanging out with them, and yes, true. So what JJ discovered was that tumor cells can downregulate T cell recognition by getting rid of ribosomal subunits. So the immunoribosome hypothesis, while it may not be generally true, it's certainly true for cancer immunosurveillance. Okay. So that's the talk until the, what was it you asked for, Soren? What was it called?
What?
Open problems, right? So everything I just told you is an open problem.
Yes.
But here are some of the more generally interesting ones. How is the law of mass action circumvented by DRIPS, physical channeling? Okay. How do immunoribosomes work? We really don't know. Are there natural immunoribosomes in non-cancer cells? We don't know. What is the efficiency of protein synthesis in living animals? It's a really important question. On the way to DRIPS, we discovered that the efficiency of protein synthesis in cultured cells, including normal cells right out of an animal, is nowhere near 100%. It's like 70 to 80%. And that's a disturbing number because protein synthesis is the single most important energy sink in a living organism. And something like 5 to 10% of the bagel you ate today went to maintaining your proteome. And any tiny selective advantage in efficiency would play out in evolution, right? Because starvation is a thing if you're an animal. Look in your backyard, they're starving, right? All day, they're just trying to eat. Okay, so what are the errors that account for this? Something that comes out of this translation work, these error numbers, which is something that my lab helped get started, is basically showing that proteins are statistical. And a 500 amino acid protein, there's a very good chance there's a change. Not a genetic change, an error, right? So proteins, you think of them as the protein. They aren't. They are they're a swarm of different protein species. They have different amino acid sequences, 1 or 2 changes. And on top of that, of course, conformationally, they're never one thing. There's a buzz. How many gene products are there really, and what do they do? What are the biological functions of degradation products of the proteasome other than immunosurveillance? There must be some, because they're really abundant. Why use alternative start codons for non-canonical proteins? Why not just always use methionine? Are there reasons? How often do they result in non-methionine initiation? Classically, even if you have a CUG, you use a methionine. A colleague of mine died, sadly, tragically, a couple of years ago. Nilab Shastri showed that sometimes you actually decode CUG, which is the most common non-cognate codon, as a leucine and not a methionine. Why? Why do you always use methionine? Why is methionine the initiating residue anyway? Something else, why are there 274 plus different human tRNAs? What the hell is going on? There's only 20 amino acids. There's only 64 codons. We only use 47 anticodons. And yet we have almost 300 tRNAs. What is going on? So many questions there. And I will end there.
Do you have any questions for John?
So it's very impressive what you're showing and what we've known for a long time from trying to model cells and create unique cells. Is almost a quarter of the genome in a minimal cell is devoted to cleaning up all the garbage in the
Yeah,
cell on
that's
a
right.
continuing basis.
That's right.
So all these systems are sloppy. Why are they sloppy? Because the binding affinities of all these things are part of a Poisson distribution and a spectrum.
Yes?
And as I said yesterday, so much of discovery is overcoming the bullshit ideas from the past. And why we ever assumed that these were high fidelity systems, it's not clear. So, but the question I have is the immunosurveillance a byproduct of the sloppiness in the ribosomes, or is, as you suggest, there's a subset of ribosomes specifically for that?
Both is what I would say. If you look at something like yeast, which don't have immunosurveillance, and there's only one paper on it, and then we did some experiments we didn't publish, and you look at the error rate in protein synthesis in terms of what's rapidly degraded. By rapid, I mean something like a half-life of 5 minutes or so. It's the same or worse than HeLa cells, right? So this looks like it's possible. You know, evolution is not about being perfect. It's just about when the tiger is chasing Craig and me, I have to be faster than him. And, right? So it just has to be good enough.
Only if we're running in the same direction.
In the same deal, fair. Or we get, yeah, as long as the tiger can't split, yes. Yeah, so I mean, it is entirely possible in the end that that's just the best evolution can do, but I would just say, in general, it's really dangerous when you have a good idea and then you have evidence supporting it. When you're a PI for a long time, these are the things that make you really scary, particularly when you're not doing the experiments yourself. And it's just very convenient to fall into what you think you know is true. And so it's just one of these things we don't know, and it's something that I think is not so easy to answer.
So one of the slides I made when I was asked to give the Schrodinger What is Life lecture, second one to him doing that, there's a number of autoimmune diseases, as you know, that are defects in the systems for taking out all the cellular garbage. And the slide was at just New York City during a garbage strike, because that's
Or
what happens
Philadelphia
inside the
normally.
cells. Yeah.
Yeah. Yeah.
What turns on the drift upon viral infection?
Yeah, good question. Right. So innate signaling really increases the peptide supply, right? And the drip we made for immunosurveillance, but there's something, this other thing which people are calling drips was not the original intention that I had. But these other defective proteins, many of which are somehow not contributing to the proteome. Yeah. So something with innate signaling actually is turning on, something Neilab discovered. Yeah. But we should sit down and talk sometime. And we're just, like neighbors. And
Okay.